All right, so welcome to Chapter 12. We're going to talk about nervous system today. So we're going to talk about, well, really from here to the end of the semester, we're going to be talking about different parts of the nervous system. In this chapter, we're going to start with, well, what are nerves? How do they function? Basically, how do they conduct signals? Things like that. And I just want to introduce you to this image here. So this is more modern type of imaging that we can do. What you're actually seeing is a picture of a nerve that is being born, actually. So what you're seeing here, colored in red, are microtubules, and green is actually actin. So that same actin that we saw in muscles and important in muscle contractions, in this case, is also going to help support and frame nerve cells. So here we have the body of the nerve cell, and then what you're seeing is that sort of the projections of nerves, the way they grow is you have a very large actin skeleton that kind of lays down the framework, and then the projection actually kind of extends out in whatever direction. It's kind of neat. And it's neat that we can actually label these things different colors so you can see the different, building blocks of cells, right? So anyway, I just thought it was kind of a neat picture. Some of the things that we've already talked about, actually seeing like hey, this is actually growing in real life. And so again, it's a snapshot in a video. So not only did they take this picture, they actually took a video watching it kind of just build itself out. All right. So let's take a look at our nervous system. All right. So in our brains, we have something like 100 billion neurons. I don't know who counted. Textbook number. A lot. 100 billion neurons, lots and lots of nerve cells. We have different cranial nerves, 1 through 12, and when we talk about them, we always use the Roman numerals. You'll see these guys in lab. There's a spinal cord and spinal nerves. We'll talk about those after this exam, as well as we'll talk about something what's known as ganglia. Again, we're going to focus on nerves, nerve functions. We'll talk more about the spinal nerves and the spinal cord later on. I've actually included it in with another chapter, with chapter 16, because chapter 16 talks about the pathways going up and down your spinal cord, and really they go hand in hand. So instead of sort of talking about one here and then talking about another one over here as if they were two separate things, as we talk about how we actually get signals from our muscles up to our brain and from our brain down to our muscles, we'll actually talk about the spinal cord then. So we're gonna talk about these things after this exam in chapters 13 and 16. Kind of put that together. Plus, it kind of cuts down on the overarching memorization you're gonna have to do as well. Which yay, that's good. Right. And then we'll end up talking about the brain, and really not too much on cranial nerves. I'll focus more on that in lab. However, we will talk a lot about the brain after this exam. Just so many things. It's going to be so cool. It's really, there's so many things, and we're only going to be able to hit, the tip of the iceberg on the brain. But it gets me really excited. It's one of the funnest parts of the semester, I think, because you finally get to take some of the stuff that you've learned and start putting it together and like, oh, how does this actually work? And you'll see all the weird things that can go wrong, and it's just a lot of fun. Okay. So anyway, before we run, you got to learn how to walk. So we're just going to learn about nerves and how they actually function, how they actually send their signals, because that'll be important in understanding how, when things go wrong, why do they go wrong. Okay. So when we talk about the nervous system, it has different functions. So it has basically a sensory function, an integrative function, and then a motor function. Okay, oops, let me go back. So when we're talking about these different functions, so sensory function is basically just that I can sense whatever it is. I'm touching, I'm seeing, hearing, right? So you actually have to be able to pick up on those cues, whatever they are, that touch cue, the light cue, the sound cue. You then have to integrate that, send it to some part of the body, usually sometimes spinal cord, usually to the brain, to then take that incoming information and give us an output. Okay, so I see light, so therefore, my eye should focus on whatever it is. That's a motor output, right? The focusing part. I feel whatever, and I should react to that, like hold it tighter, hold it less tight, whatever. So again, you have a sensory function, sends that information to some part where you're going to take that sensory information and process it and turn it into a motor output. When we talk about our nervous system, we have two major subdivisions. The first is the central nervous system, which is the brain and the spinal cord, and then the peripheral nervous system, which is basically all of our nerves. So cranial nerves, basically coming straight off the brain. Spinal nerves coming off the spine. Something called a ganglia, we'll talk about that in a second. You actually have a lot of nerves in your intestines as well. Something like, I forget the exact number, but something like 1 million different nerves in our intestines. They often say that the gut, there's like a second brain in the gut because of all the neurons that are located there. And you can see in the peripheral nervous system also lots of sensory receptors in the skin, and we'll talk about those as well. And hide me again so you can get a better view of things. But again, let's start at what is actually a nerve. What are the parts of an actual nerve? All right, so another name for nerve is a neuron, and that's really how I will be talking about them from here on out. I'll be calling them neurons. Now, it has sort of the usual parts plus, sort of unique parts to it because of its unique function. So it'll have a cell body, and we call this either the cell body, another name for it is a perikaryon, or sometimes you'll see it referred to as the soma. Any of those terms are okay. In the cell body, you would find all of the usual organelles, so rough endoplasmic reticulum, smooth endoplasmic reticulum, mitochondria, Golgi complex, all of this, plus something, so again, all the normal things, lysosomes, all that, plus something called a nissl body. I should say there's plural, there's many of them. And basically what a nissl body is free ribosomes and clusters of rough endoplasmic reticulum. You guys remember what ribosomes do? Hopefully you remember that they make proteins, right? So let's think about this for a second. These cells, these neurons, have lots of extra ribosomes. Why? Why would they need so many extra ribosomes? What proteins are they making? Give up. Neurotransmitters. So all those neurotransmitters I'm sure you guys have heard of, dopamine, serotonin, epinephrine, all of those are proteins. Cells have to make lots and lots and lots of them. So to make that, they have extra ribosomes and even more rough endoplasmic reticulum than other cells do. Okay. They'll also have things called neurofibrils. These are sort of the same things, the cytoskeleton, these give the cytoskeleton the shape and support to the cell. And similarly with microtubules giving shape support. Plus, as we had mentioned like way back at the beginning of the semester, microtubules will form sort of highways that then vesicles containing compounds, in this case neurotransmitters, will be trafficked down these microtubules from the cell body down the length of what's known as an axon on the cell, before it actually ends up leaving the cell. I think we kind of know this intuitively, right? So if the cell is making all of its neurotransmitters here, it doesn't just release it haphazardly. It doesn't just all kind of come out of the cell here. What's going to happen is that cell is going to extend this long appendage down to wherever that point is, what other tissue it wants to communicate with, and then those neurotransmitters get released down at this end. Okay. We have something known as the dendrite on cells, and depending on the type of neuron it is, it'll have slightly different types of dendrites on it. So in this case, you can see all these kind of look like wild hairs on the soma. Those are all dendrites. So dendrites, let's see, they take, it's terrible handwriting, take incoming information to the soma or cell body. Okay, so dendrites will get information from somewhere, either from the environment, from another cell. They're going to take that information and take it to the cell body. And then in that cell body, maybe you'll increase the amount of neurotransmitter you're making, or you're going to transmit that information further on, but something's going to happen, okay? Excuse me. Okay, so in the axon then, this long stalk coming off of the dendrite is going to be the axon. So the axon, see how well I can write here. So the axon is going to transmit outgoing information to another neuron, cell, muscle, or tissue. Okay, so basically the axon is going to take all this information, it's going to travel down the length of this axon, and then it's going to take that information, and then it's whatever it's making contact with, just depending on where you are. Like if you're in your brain, the neuron might contact another neuron. If you're a different body part, it might contact a cell or a muscle or some other tissue. So it just depends on what it's making contact with. Okay, so, but it takes that information and sends it somewhere else. When we classify neurons, what we're going to talk about is we can either structurally classify them or functionally classify them. And by now you should kind of get used to this idea that we can either do things based on what they look like or based on how they work. So when we talk about structural classification, they can either be multipolar, bipolar, or unipolar. And we're going to see an image of this in a second, and it's easier to explain those when you're actually looking at a picture of them. So I'm going to hold off on that explanation. Or we can classify them by their function. So again, this is either sensory motor or something called an interneuron. This is what I was mentioning to you before. So sensory neurons are going to take information from wherever, their environment, so their external environment or internal environment. They're going to take that, they're going to transmit that to what are known as interneurons or association neurons, and then those neurons are going to take that information and give you a motor output. So that's outgoing information, it's efferent. So again, I touch something, I either, if it's, this pen, I'm touching it, that information's going up to my brain, I'm going, okay, let me motor output, this is how strongly I need to hold this pen. I don't need to squeeze it super hard, not too light because then it'll fall out of my hand. That's a motor output. And again, I have to take that sensory, convert it into motor. So that's the job of that interneuron, converting one into the other. Let's try to get this advanced. All right, so here we're seeing our structural classifications. So this is what I was mentioning to you earlier. It's just a little easier to show you. And I think I'll kind of go right to left, because again, I think it's a little bit easier to do it that way. So let's take a look at this one, the unipolar neuron. So here we have the cell body, and if you'll notice, there's only one thing coming out of that cell body, and then that one thing will split into either the axon or the dendrites, right? So it's unipolar because only one thing comes out of the cell body. Right? In the case of something that's bipolar, you'll see that there's one stalk coming out here on the top, one thing coming out here at the bottom. The one on the top will eventually split into many dendrites. The one on the bottom will split into many axon terminals. However, only two things are coming out of the cell body. It's bipolar. Okay? And then in this one, we see there's one axon coming out, many dendrites coming out of the cell body itself. So it's multipolar. It says many. So that's the structural classification. You either see something that looks like this, this, or this. All right. I think I might have mentioned this to you guys in the lab, but if not, let me say it now. Neurons are so specialized in sending these signals and receiving signals, interpreting those signals, and sending them along their way, that they are very bad at taking care of themselves. So they have to have specialized cells, and we call those neuroglia cells, to take care of them, to help isolate them from potential harmful substances in the bloodstream, to helping them recycle neurotransmitters, to a whole bunch of other things, to other cells having to clean up after them. Okay, so all of that is a job of the neuroglia. They're going to help take care of the nerve cells. So the neurons. So we have 4 different types of neuroglia. We have astrocytes, oligodendrocytes, microglia, and something called an ependymal cell. And you can kind of see them in this image. So let's talk about astrocytes first. You can see here, these are fibrous astrocytes. Right here, here's a protoplasmic astrocyte, just a slightly different shape to it. But one of the things you'll notice is that it has an appendage touching that blood vessel, and then it's also going to be touching the neuron in some way, right? So what this is going to do is it's going to support the neuron, but the other thing it's doing is it's isolating the neuron from harmful substances in the blood. So basically what's going to happen is that in the bloodstream, right, that's where we have all our nutrients. So what it's going to happen is this astrocyte is going to pick up those nutrients and transmit them to the neuron. Okay, so basically it's going to feed that neuron. It's going to regulate the ion transport. It'll regulate what can get through this blood vessel as well. All right. So it also will regulate the growth, the migration, the interconnections of neurons. So all of that, it helps modulate all those different things. Again, ion transport. If that neuron releases excess neurotransmitters that are not needed for cell communication, it will pick up after that neuron, if you will. It'll take up those excess neurotransmitters, and a lot of times what it'll do is it'll either break them down or it'll simply give it back to the neuron. So it kind of shuttles them back, like here, you lost these, here they are. Okay, so those are the astrocytes. Really important little helpers for neurons. Come on. Let's see. Get my slide to advance, and it's not wanting to work with me. All right, the next type of cell we're going to take a look at is something called an oligodendrocyte. And these guys are unique. They're kind of interesting in what they do. They have a cell in the peripheral nervous system that's also going to do a very similar function. They are a different cell, have a different name, but basically oligodendrocytes are going to share their function. In any case, so we're in the central nervous system, so that means we're looking at things in either the brain or the spinal cord. So what these guys are going to do, they kind of look like, they remind me of little octopuses. So here's an oligodendrocyte. Can you see how it's basically sending this arm, if you will, and it's wrapping itself around this axon? Okay. Well, basically that wrapping that it's creating is called a myelin sheath. Okay, and this myelin sheath is going to insulate that axon, sort of like you insulate wires in your house. Right? You don't have bare wires running everywhere. You have that plastic rubbery coat around them. So it's going to insulate them, and by doing that, it's going to increase the speed of nerve impulses. Right? It's really important, and we'll talk a little bit later on about how that actually, why that actually works that way. We're going to see there's other cells called microglia cells. So here's a little microglial cell right here. There's one over here. And basically these microglia are phagocytes. So that means that they're going to gobble up things that don't belong. So sometimes damaged tissue, any kind of microbe, hopefully it'll gobble those up. Obviously they don't always get it, and you get infections and things like that. However, you do have phagocytes there, kind of cleaning up the space that neurons are in. And then finally you have something called ependymal cells. Okay. These are going to contribute to the blood-brain barrier. They're forming the blood-cerebrospinal fluid barrier. So basically these cells are producing the actual cerebrospinal fluid. So what's going on is they're going to take material from the bloodstream. They're essentially going to filter certain things out of the bloodstream, and they create that cerebrospinal fluid. Now, cerebrospinal fluid, if you ever get a chance to see that, you can even just do a Google video on a spinal tap. It is incredibly clear, like crystal clear fluid. And that's because a lot of the elements from the bloodstream have been filtered out. And basically what these ependymal cells are trying to do is create the perfect environment for the neurons so that they can generate the signals that are necessary for their proper function, right? So again, these neurons are very sensitive to environmental disruptions. So the cerebrospinal fluid is there to help make sure that you have the right ion balance, fluid balance, to make sure that your brains and nerves are functioning correctly. And this last little point here, they contribute to the flow of cerebrospinal fluid. You'll see that they have some cilia on them. And again, wherever you have cilia, you're going to have movement. So the beating of the cilia helps the cerebrospinal fluid sort of flow around in the spaces where we find it. And we'll talk more about that later. Okay. In the peripheral nervous system, so now we're talking about outside the brain and the spinal cord, we also have some neuroglial cells. Not as many, just 2 different types. We have what are known as Schwann cells, and these Schwann cells are going to be analogous to those oligodendrocytes that we found in the central nervous system. So here's this Schwann cell, and what we'll see is that it'll take itself and it'll simply wrap itself around the axon. And remember, it's a cell, so it's going to have that phospholipid bilayer. So as it wraps itself, basically you end up having this sort of fatty wrapping around that axon, which acts as a very nice insulator. Okay, so that wrapping is called the myelin sheath. Okay, and that wrapping is going to help with the generation, or I should say, the speed of the nerve signals. Okay. And then we also have satellite cells, and these satellite cells are going to help maintain the cell bodies of nerves in the peripheral nervous system. Okay, so you can kind of see they're helping protect and support the cell body. And here's just an image of that Schwann cell wrapping itself. So it would start off looking like this. You can see it wraps, keeps wrapping itself, and as it wraps itself tighter and tighter, all those things that you'd find in the cytoplasm get squeezed to the outermost layer, so the nucleus and everything else, until you have this nice wrapping of this phospholipid bilayer all the way around, and that's what produces the myelin sheath. So the wrappings, again, are the myelin sheath. The cell is a Schwann cell. The outer layer, the outer wrap, where you see all the cytoplasm, you see the nucleus and everything, that's called the neurolemma. You'll come across this term in lab if you haven't already. All right, just a few more definitions, and then we'll conclude this video. So what we're going to find is that depending on where you are, whether or not you're in the central nervous system versus the peripheral nervous system, the same things will get called different things. And it's not, really the terms in this case are not interchangeable. So a ganglion is a cluster of cell bodies, neuron cell bodies, in the peripheral nervous system. That same thing, the cluster of neuronal cell bodies, we're in the central nervous system, we call that a nucleus. Okay, and don't get confused between, well, each cell has its own little nucleus. Yes, you're right. But we reuse that term, and I'm not exactly sure why, but they reuse that term to mean a cluster of actual cell bodies. Okay, is also a nucleus in the central nervous system. A bundle of axons in the peripheral nervous system, we call those nerves. When we're in the central nervous system, we call them tracts. And so again, the name changes, and they're not interchangeable. You do not have a nerve in your spinal cord. You do not have a nerve in your brain. You have tracts. You do not have tracts in your peripheral nervous system. You have nerves. Okay. Basically, when we see things and they look white to us, these are going to be myelinated axons. They look white because of the fatty phospholipid bilayer that they're wrapped around. The gray matter is going to look gray because they don't have myelination, essentially. So these are things like cell bodies do not have myelination, dendrites do not have myelination, obviously unmyelinated axons do not have myelination, and those neuroglia cells also do not have myelination. So that's where we would see gray matter. So when we see something gray, it'll be one of these things that we are looking at. All right. So we're going to stop there. We'll pick up from there in the next lecture. Hopefully that made sense to you. As always, feel free to email me with any questions, and I will see you next time.